Good morning, everyone. I am delighted to welcome you all today. My name is Dr. S.K. Singh, and I am pleased to have the opportunity to talk with you about an important aspect of our work. Today, we will explore the sources of contamination in cleanrooms and how we can control them. This presentation will cover the common contamination sources, their impact, and effective control measures. I look forward to sharing valuable insights with you all. Let's get started. Now, let's take a closer look at the different types of contamination that we need to be aware of in cleanroom environments. First, we have particulate contamination, which includes dust and fibers. These tiny particles can easily become airborne or settle on surfaces, potentially compromising product quality. Next, microbial contamination involves bacteria and fungi, microorganisms that can grow and cause serious safety concerns. Then, there's chemical contamination, which includes vapors and residues from chemicals used in manufacturing processes. These can be invisible but highly damaging. Lastly, we have cross-contamination, which occurs when a product transfers contaminants to another product, either directly or indirectly. It's important to understand that cleanrooms are specifically designed to control and minimize these types of contamination. They are used across various industries like pharmaceuticals, biotechnology, semiconductors, and healthcare, where the integrity and safety of products are critical. Contamination isn't just an inconvenience. It can seriously affect product quality, safety, and regulatory compliance. So, maintaining strict control over these contamination types is fundamental to ensuring that we meet the high standards required in these industries. Let's take a closer look at cleanroom classification. First, we have the ISO 14640-41 standards, which categorize cleanrooms from Class 1 to Class 9 based on particle counts. These classifications help ensure that the environment meets specific cleanliness levels, depending on the application. For ultra-clean environments, like those in semiconductor manufacturing, ISO Class 1 is used, while less critical environments might be ISO Class 9. Next, we have the GMP grades, which are used primarily in pharmaceutical and biotech industries, especially within the EU and WHO guidelines. Grade A is the most critical zone, typically where aseptic filling takes place, so the environment must be extremely controlled to prevent contamination. Grade B serves as the background environment for Grade A, providing a cleaner surrounding area. Grades C and D are less critical, used in stages that don't require as stringent control, but still need to maintain cleanliness standards. Both the ISO classifications and GMP grades define specific limits for particle counts and microbial contamination. These limits are crucial because they set the standards to keep the environment safe and suitable for sensitive processes. So, understanding and properly implementing these classifications ensures we maintain the integrity and safety of our products, connecting directly to our overall goal of quality assurance. Let's talk about the source of contamination, focusing on the major contributor, personnel. Humans are responsible for approximately 80 to 90% of contamination in many controlled environments. This is mainly due to things like skin flakes, which can release around 1 million particles per minute, as well as hair, sweat, and respiratory droplets. For example, poor gowning practices can lead to microbial contamination, especially during sensitive processes like aseptic filling. To control these risks, we implement strict measures such as sterile gowning, including coveralls, gloves, and masks, to create a barrier. Equally important is comprehensive training and adherence to standard operating procedures. Additionally, controlling movement and behavior within the clean environment helps minimize the chances of contamination. All of these measures are essential because, ultimately, personnel are often the most significant source of contamination, and managing this risk is key to maintaining a sterile environment. Now, let's continue exploring the sources of contamination, specifically focusing on equipment as a potential source. Equipment can generate particles through various mechanisms such as friction, vibration, wear, and tear. Over time, these processes cause parts of the equipment to shed particles, which can contaminate the manufacturing environment. For example, in tablet manufacturing, small particles might come off equipment parts and end up in the product. To control this, we emphasize the use of non-shedding materials like stainless steel 316, which is known for its smooth surface and durability. Regular preventive maintenance and calibration are also crucial to ensure equipment functions properly and doesn't produce excess particles. Additionally, equipment qualification, covering installation, operational, and performance qualification, or IQ, OQ, and PQ, is vital. These steps ensure that equipment is installed correctly, operates within specified parameters, and consistently produces quality products without contributing to contamination. So, by selecting the right materials, maintaining equipment diligently, and verifying proper operation through qualification, we can significantly reduce contamination risks associated with equipment in our manufacturing processes. Now, let's continue by looking at the source of contamination, specifically materials and consumables. These are often overlooked, but they can be a significant source of particles in the environment. For example, packaging materials like cardboard boxes can release fibers into the cleanroom. Similarly, raw materials themselves can introduce particles if not properly managed. To control this, we need to focus on using materials that are compatible with cleanroom standards, materials that don't shed particles or fibers easily. Implementing pass boxes and material airlocks is also crucial, as they help transfer materials in and out without contaminating the environment. Additionally, surface disinfection before bringing materials into the cleanroom is a vital step. It ensures that any particles or microbes present on the materials are eliminated before they can compromise the environment. Remember, controlling the sources of contamination like materials and consumables is essential for maintaining the integrity of the cleanroom and ensuring product quality. Now, let's continue our discussion on sources of contamination, focusing on HVAC and airborne contamination. As you know, air is the primary carrier of particles that can carry contaminants, making it a critical factor in maintaining a sterile environment. The main sources of airborne particles include external air, which can introduce contaminants from outside the controlled space, and poor filtration systems that fail to adequately remove particles from the air. To control this, we rely on several key technologies. HEPA filters, for example, are highly effective, capturing at least 99.97% of particles as small as 0.3 micrometers. ULPA filters go even further, with efficiencies of 99.999%. Laminar airflow systems are also essential. They create a unidirectional flow of filtered air that helps sweep away contaminants from critical areas. Additionally, maintaining appropriate air change rates, ranging from 20 to 600 air changes per hour depending on the specific class or environment, is vital to ensure that contaminated air is constantly replaced with clean, filtered air. All these measures work together to minimize airborne contamination, which is essential for protecting both product quality and patient safety in our sterile environments. Let's continue by looking at how airflow and pressure control play a crucial role in contamination prevention. In critical zones, we often use unidirectional, or laminar, flow to ensure that air moves smoothly and consistently in a single direction. This minimizes turbulence and helps carry away contaminants effectively. Now, pressure differentials are also vital. We maintain higher pressure in cleaner areas compared to surrounding spaces. For example, in pharmaceutical sterile rooms, we typically keep a pressure of +10 to +15 pascals. This positive pressure acts like a protective barrier, preventing contaminated air from entering these sensitive environments. Controlling these factors involves proper airflow design, making sure the air moves correctly through the space, and continuous pressure monitoring systems. These systems are essential because they provide real-time data, alerting us immediately if pressures fluctuate or if airflow isn't maintained properly. Overall, by managing airflow and pressure carefully, we create a safer, cleaner environment that minimizes contamination risks and supports the integrity of our sterile processes. Let's continue by discussing the sources of microbial contamination. As you can see, personnel, water, and surfaces are common contributors. These are the main pathways through which microbes can enter and contaminate our environment. Some of the most common microbes involved include Staphylococcus, Bacillus, and fungi. These microorganisms can pose significant risks if not properly controlled, especially in sensitive settings. Now, how do we control this contamination? Routine cleaning and disinfection are essential. They help to eliminate or reduce the microbial load regularly. Using sporicidal agents is also important, particularly when dealing with spores that are more resistant and harder to eradicate. Environmental monitoring plays a crucial role too. Techniques like settle plates and swabs allow us to detect any microbial presence in the environment. This ongoing monitoring helps us identify potential problems early and maintain a safe and clean environment. All of these strategies are interconnected and vital for minimizing microbial contamination, which ties back to our overall goal of ensuring safety and quality in our operations. Let's talk about cleaning and disinfection, which are absolutely critical for controlling contamination in our facilities. Ensuring a clean environment helps protect both products and personnel. Now, for methods, we typically use wet mopping and wiping techniques to physically remove dirt and microbes from surfaces. These methods are quite effective when done properly. As for the agents we use, alcohols like 70% isopropyl alcohol are common because they're fast-acting and effective against a broad range of microorganisms. Hydrogen peroxide is another powerful disinfectant that's often employed, especially because it breaks down into harmless byproducts, making it environmentally friendly. Now, from a regulatory standpoint, it's important that our cleaning procedures are validated, meaning we have proven that these procedures consistently achieve the desired level of cleanliness. Additionally, rotating disinfectants is a key strategy to prevent microorganisms from developing resistance. This rotation ensures we're not relying on just one disinfectant over time, maintaining the effectiveness of our cleaning protocols. Connecting this to the overall theme, rigorous cleaning and disinfection are fundamental pillars in maintaining a safe, compliant, and contamination-free environment. Let's now take a look at facility design, specifically focusing on monitoring and validation. First, for non-viable monitoring, we typically use particle counters. These devices help us keep track of airborne particles that could affect product quality. For viable monitoring, we rely on methods like settle plates and air samplers, which allow us to culture and identify actual microorganisms present in the environment. The frequency of these tests is usually determined by Good Manufacturing Practice, or GMP, guidelines to ensure consistent environmental control. Moving on to validation, two key tests are essential. The HEPA integrity test, often called the PAO or DOP test, verifies that our HEPA filters are functioning properly and effectively removing contaminants. Additionally, airflow visualization using smoke studies helps us confirm that airflow patterns are correct, ensuring a clean environment. Now, control measures are equally important. We strive to have smooth, non-porous surfaces throughout the facility because these are easier to clean and disinfect. Rounded corners, or coving, are used to eliminate hard-to-clean gaps and prevent dust accumulation. We also incorporate airlocks and gowning rooms to control contamination from personnel entering and exiting clean areas. As an example, having a separate entry and exit point for personnel or materials can significantly reduce the risk of cross-contamination. Everything in facility design and validation ultimately aims to protect product integrity and patient safety, aligning with the overall goal of maintaining a controlled, compliant environment. Let's take a closer look at some of the key regulatory guidelines that govern our cleanroom practices. First, we have the ISO 14644 series, which sets the international standards for cleanroom classifications and control. These standards help ensure consistent cleanliness levels across different facilities and industries. Next, there's the WHO GMP guidelines, which provide comprehensive directives for pharmaceutical manufacturing quality standards worldwide. They emphasize the importance of environment control, documentation, and personnel training. Then, we have the EU GMP Annex 1, which was recently updated in 2022. This annex specifically details the requirements for sterile manufacturing, highlighting the importance of maintaining aseptic conditions and rigorous validation procedures. Lastly, the US FDA guidance for aseptic processing offers detailed recommendations for sterile drug manufacturing in the United States. It underscores the importance of risk assessments and environmental monitoring. Across all these guidelines, some key requirements stand out. Proper documentation is crucial, as it ensures traceability and accountability. Well-written standard operating procedures, or SOPs, are essential for consistency and compliance. And of course, thorough training and regular audits help maintain a high standard of practice and quickly identify any areas needing improvement. All these elements come together to support our overall goal, ensuring patient safety through impeccable sterile manufacturing practices. They form the backbone of a robust quality system that keeps our processes compliant and our products safe. Let's wrap up with some key takeaways. Cleanroom contamination mainly comes from four main sources: personnel, equipment, materials, and air. Understanding these sources helps us target our control efforts effectively. To manage and reduce contamination, we need a combination of controls. Engineering controls like HVAC systems and HEPA filters are crucial. They help create and maintain a clean environment by filtering out particles and microbes. At the same time, administrative controls such as standard operating procedures and staff training are essential. These ensure everyone knows how to work properly within the cleanroom and follow best practices. Finally, it's important to remember that compliance with these controls isn't just about following rules. It directly impacts product quality, safety, and regulatory approval. When we get this right, we're not just maintaining a clean environment, we're ensuring the integrity and success of our products. For further support, please don't hesitate to reach out. If you have any questions or need additional information, we're here to help. Feel free to contact us through the provided channels. Thank you so much for your time and attention today. We truly appreciate your engagement and look forward to assisting you further.